This is LATHA AKULA. In the last videos, we have completed the introduction to biomolecules. We have studied about what are the different types of biomolecules and what are the primary and secondary metabolites. So in this video, we are going to study about individual biomolecules, and that the first one is the carbohydrate. So carbohydrates, they were generally called as saccharides because the main component in this is the sugar. Hence, the carbohydrates were called as commonly saccharides. And what are the components present in it? They consist of carbon, hydrogen and oxygen. Carbohydrates are made up of carbon, hydrogen and oxygen, and they were present in the ratio of 1:2:1. That means if it consists of one carbon and it has two hydrogens and one oxygen. And the general formula of this is CnH2nOn is the general formula of carbohydrate. And the carbohydrates are also called as hydrates of carbon. And if you see the carbohydrates, the classification of the carbohydrates, there are two types of carbohydrates were present. They were small carbohydrates and large carbohydrates. The small carbohydrates will study under biomicromolecules. We have studied about biomicromolecules and biomacromolecules, and which will be included under acid soluble pool. These biomicromolecules will come under acid soluble pool, and whereas biomacromolecules will come under acid insoluble pool, where we have studied about the chemical analysis of the living tissue. And here the carbohydrates can be classified into small and large carbohydrates, and these small carbohydrates will be under the biomicromolecules, will be studied under biomicromolecules because they have the less molecular weight. And these small carbohydrates again divides into monosaccharides, derived monosaccharides and oligosaccharides. Monosaccharides, derived monosaccharides and oligosaccharides. So monosaccharides, they have only one sugar, and derived monosaccharides and oligosaccharides. If you see, the first one is the monosaccharides. The name itself indicates they have only one sugar. And here, these monosaccharides are also called as simple sugars because they has only one sugar. Hence, what is another name for the monosaccharides? They are also called as simple carbohydrates. And these monosaccharides, they have only one sugar, and those sugars, they may have 3 to 7 carbon atoms. How many carbon atoms may present in the monosaccharides? Some of the carbohydrates, they consist of 3, some have 4, some have 5, 6, 7, like that. 3 to 7 carbon atoms were present in the monosaccharides. If the carbohydrate that consists of 3 carbon, then it is called as triose sugar, and the formula is C3H6O3, and example is glyceraldehyde. And if the carbohydrate has 4 carbon atoms, then it is called as tetrose sugar, and the formula is C4H8O4. C4H8 and O4. An example, erythrose sugar. And 5 carbon atoms, if it has 5 carbon atoms, it is called as pentose sugar, and its formula is C5H10O5. An example are ribose and arabinose. And if it has 6 carbons, it is called as hexose sugar. Generally, we will study about hexose sugar, right? Its formula is C6H12O6. And examples for this hexose sugar is glucose, galactose and fructose. And if it has 7 carbons, it is called as heptose sugar, and its formula is C7H14O7. Example are pseudoheptulose. These are the several examples of different carbohydrates depending upon the carbon atoms, and these examples are very important. The names of these examples are very important. So of these, the most important one is hexose sugar. In that hexose sugar, we will see one example. And if these carbohydrates, they may have the functional group, either aldehyde or ketone group may be present. Aldehyde means CHO group, ketone means C double bond O group. Some aldehydes, some carbohydrates, they have aldehyde group. Those sugars are called as aldose sugars. And some carbohydrates, they have ketone group as a functional group, and that those sugars are called as keto sugars, and examples for the aldose sugar is glucose, and where the functional group is aldehyde, that is CHO, and the example for the keto sugar is fructose, where the functional group is keto, that is C double bond O. In that, the one example we'll study, that is glucose. What is the formula of the glucose? C6H12O6. It is an aldose sugar, that means it consists of aldehyde. And if you see the structure of a glucose, this is the open chain structure of the glucose. It consists of 6 carbon atoms. How many hydrogens? 12 hydrogens and 6 oxygens were present. And its functional group is aldehyde group. And from this open chain, from the 1 carbon atom and 5th carbon atom, one water molecule is removed. Then it will form the closed chain structure. Here what will happen? From 1 carbon and the 5th carbon, H plus OH will be removed in the form of H2O, and this H2O will dissociate into H and OH, which will be joined in the first carbon, and it forms this ring-like structure. This ring-like structure, if it is elaborated, it will form a hexagonal ring, which is also called as pyranose ring. And this is about the hexose sugar, example glucose. The second monosaccharide is derived monosaccharide. Here, what are derived monosaccharides? Some monosaccharides, they were modified and formed the other substances. They were called as derived monosaccharides. In that, the first one is deoxy sugar. Deoxy sugar is, for example, if we have taken ribose, which is a 5 carbon compound pentose sugar, it has this structure. And in this structure, in the second carbon atom, if there is a hydroxyl group is present, right? In that, from this second carbon, oxygen was removed, then it forms a deoxyribose sugar. So it means deoxygenation of the ribose sugar, that form the deoxyribose sugar. That means from the ribose sugar, deoxyribose sugar was formed. And here this ribose sugar is present in RNA, and deoxyribose sugar is present in the DNA. So this is about deoxygenation. So what is happening here? From the ribose, the second carbon, there is a hydroxyl group is present. From that, the deoxygenation and removal of oxygen, that resulting in the formation of deoxyribose sugar. This ribose sugar is present in RNA, and deoxyribose sugar is present in the DNA, the nucleic acids. RNA consists of ribose sugar and DNA consists of deoxyribose sugar. And this another one is the amino sugar. Amino sugar example is glucose amine, which is present in chitin. It is present in the cell wall of the fungi. And sugar acid. Sugar acid example is glucouronic acid and ascorbic acid. And sugar alcohol. Sugar alcohol is nothing but example is mannitol. This is present in the brown algae. These are the some of the derived monosaccharides, and all these are the biomicromolecules only. Next, third one is the last one in the simple sugars is the oligosaccharides. Last one in the small carbohydrates is the oligosaccharides. Here, oligosaccharides are formed by the condensation of 2 to 9 monosaccharides. Here, how many monosaccharides? The condensation of 2 to 9 monosaccharides that form the oligosaccharides. So what is the condensation? It is a chemical reaction. Condensation is a chemical reaction where 2 monosaccharides were joined by the removal of water molecule. How it is formed? For example, this is one sugar and this is another sugar. Between these two, there is a removal of water or a condensation process takes place, and these two get joined. That is called as condensation process. And here, of course, they consist of 2 to 9 monosaccharides. These are also biomicromolecules only. And how these monosaccharides have been joined? By means of glycosidic bond. How these monosaccharide units have been joined? By means of glycosidic bond. And how this glycosidic bond is formed? For example, we have already studied these sugars. They consist of aldehyde or ketone functional group. Either aldehyde or ketone of one monosaccharide is connected to the alcohol group of other monosaccharide that form the glycosidic form. How the glycosidic bond is formed? Either aldehyde, if it is aldo sugar, they consist of aldehyde group. If it is a keto sugar, they consist of ketone group. Either aldehyde or ketone group of one monosaccharide is connected to the alcohol group of other monosaccharide, then it forms the glycosidic bond. Not only this, but also the dehydration process also form the glycosidic bond. How? Between the first carbon and the fourth carbon of monosaccharide. See here, this is the first carbon and this is the fourth carbon. There is a dehydration process takes place. Here OH group is present, here also OH group is present. Then what will happen? From this H2O was removed. That is, dehydration process occurs and resulting one is glycosidic bond. Like this, the glycosidic bond is also formed by the dehydration process or removal of water between the first carbon of one monosaccharide with the fourth carbon of another monosaccharide. Glycosidic bond. If it is alpha sugar, they form the alpha 1,4 glycosidic linkage. If it is beta sugar, they form the beta 1,4 glycosidic linkage. And here, these depending upon the number of monosaccharides condensed, these oligosaccharides are different types. Here we have studied that how these oligosaccharides are formed by the condensation of the monosaccharides by the forming the glycosidic linkages. The oligosaccharides are formed. Depending upon the number of monosaccharide units that have been condensed, there are disaccharides, trisaccharides and tetrasaccharides. Disaccharides means what? If two sugars have been condensed and by means of glycosidic bond, they have connected, that is called as disaccharide. They consist of only two sugars. Examples: sucrose, maltose, lactose, sucralose are the examples for the disaccharides. See here, this is the maltose. This is the maltose. How this maltose is formed? By the condensation of the two glucose units. Two alpha glucose units condensed and form the disaccharide called as maltose. And how the condensation process occurs? By the removal of water molecule from the carbon 1 and from the carbon 4 of another monosaccharides. There is hydroxyl groups are present. Those hydroxyl groups are removed removed in the form of water molecule and form a glycosidic linkage between them. And this is maltose. And how the sucrose is formed? Here the sucrose is formed by the condensation of the glucose and lactose. Glucose and fructose. And lactose is formed by the condensation of the galactose and glucose. Like this, different disaccharides were present. Next, trisaccharide. If three sugars have been condensed, they form the trisaccharide. Example for the trisaccharide is the raffinose. Here, what are the sugars condensed? Glucose, fructose and galactose. They condense and form the raffinose sugar. Trisaccharide. And tetrasaccharide. Tetrasaccharides are formed by the condensation of the four monosaccharides. Examples: stachyose. This stachyose sugar is present in the artichoke and soybean. And what are the sugars involved in the formation of this tetrasaccharide? Two alpha D galactose units, one alpha D glucose unit and one beta D fructose units. They condense and form the stachyose sugar. Like this, different sugars are different depending upon the number of monosaccharide contents. We have different oligosaccharides. Reducing sugars. So the reducing sugars are the sugars which reduce cupric ions to cuprous ions are called as reducing sugars. And this property is the basis for the Benedict and Fehling test. So this test is done in the laboratory to identify whether the sugar is present in the urine or not. And here this property is found in sugars which have free aldehyde and free ketone group. This is seen in almost all monosaccharides. There is a free aldehyde and ketone group is available. Hence almost all monosaccharides are the reducing sugars only. But in disaccharides there is one exception, that is in the case of sucrose. The sucrose is a disaccharide which is made up of glucose and fructose. As we know, glucose is a aldose sugar and fructose is a ketose sugar. Here, between the two monosaccharide, there is a glycosidic linkage is formed, right, in the disaccharides. How this glycosidic linkage is formed? Here, the aldehyde group of glucose and the ketose group of fructose, they both have involved in the formation of glycosidic linkage. That means there is no free aldehyde or ketone group is available. Hence, sucrose is called as non-reducing sugar because there is no free aldehyde or ketone group is available. Both aldose and ketose sugars have been involved in the formation of glycosidic linkage. But whereas other disaccharides like lactose and maltose, there is a free aldehyde or ketose groups are available and hence they were called as reducing sugars. This is about the property of reducing sugars. With this we have completed the small carbohydrates. In the next video we'll